Share my screen. Can you see mitosis and meiosis?
Yes.
Okay. All right, so this is where I'd like to start. I'm going to review the entire topic.
Can you zoom in though? I think we can only see it from far away.
Okay. I'd like to leave it like this for right now. I will use it later, the slideshow. Okay, can you read that? Mitosis, meiosis, okay. So why do we have mitosis and meiosis? They only occur during cell division. And we need mitosis in order for organisms to grow and develop and reproduce. And we need meiosis to introduce genetic variation. And we'll come back and reiterate those points. What I'd like to do first is actually stop sharing this.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Excuse me.
Yeah.
Apparently you have to let someone in. Somebody's getting the waiting room again.
Oh my gosh, I thought I had that problem solved. Okay. I don't see anybody in the waiting room.
Oh. All right, I'll let him know.
Yeah, and if they need the passcode, you've got that, right, James?
Yeah.
I'll tell them that if they need to. Okay. All right, I want to do another video. That one was on mitosis. I want to do another one by comparison with meiosis. This is the amoeba sisters.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn out cells, but not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes. And your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. If you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes because the sister chromatids are still attached and we're actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. A little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. Because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They're actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes hop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful as having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes and the ending cells only had 23. So they are not identical to the original and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Neva Sisters, and we remind you to stay curious.
Okay, any questions about either of those videos or about the slide set from last time? That slide set is in Canvas, and I hope you've all had a chance to review it before this class. Are there any questions about last time? I can page through the slides if you like, but if there are no questions, I'll just proceed with the worksheets. Maybe it would be useful if
Could we go over meiosis?
yeah, I will. So let's share the screen again. All right. I'll flip through the slides one by one, and you can stop me if you wish. Karyotypes, remember trisomies, mitosis, and again, the sole purpose of mitosis is to produce more cells, identical to the parent and identical to each other. And the reason we need mitosis is to make more cells for growth and to repair any damage that ever gets done, and to allow for cell specialization. So you remember when you start out with a zygote, you have one cell that is diploid, so it has all the genes and the right number of copies, but it's just one cell completely undifferentiated. And you end up, in the case of humans, with a complex body that has over 200 different cell types. So as mitosis occurs and you keep getting more and more cells, these cells can respond to stimuli and differentiate. So they can differentiate into skin cells, foot cells, fingernail cells, and so on. So mitosis allows for that. The other important thing to remember about mitosis is that there is no genetic variation. In fact, it's the opposite. The goal of mitosis is to be high fidelity, to have absolutely no changes between the parent and the daughter cells. So that's...
Professor, can you repeat that last part one more time? You said it's important.
Yeah, it is important that there's no genetic variation in mitosis. And we're going to contrast that with meiosis. But the goal of mitosis is no change. It's conservative. Mitosis wants the children, the progeny, to be identical with the parent and identical with each other. So there's no change, no genetic change. There are regulatory changes that can occur to give cell specialization, but there are no genetic changes in mitosis. All right, so then we contrast that with meiosis. So what are the consequences of meiosis? It starts with a diploid cell, just like in mitosis, but it ends up haploid. It ends up with half the number of chromosomes that it started with. And it achieves that by going through not one, but two cell divisions. So the chromosome number is reduced by half from 2n to 1n. And a point I want to emphasize is that meiosis generates genetic variation. It produces it. A main goal of meiosis is to produce genetic variation. So in contrast to mitosis, where the goal is to keep everything the same, the goal of meiosis is to change everything. Make sure that the progeny that are produced are unlike the parent and unlike each other. And the video that we just looked at, the amoeba sisters, made a big point of that. And why is that so important? It's because species could not evolve without genetic variation. So natural selection, as environmental conditions change, and environmental conditions call for new types within a species, natural selection needs something to select from. So if all it had was mitosis, it would keep selecting from the same pool over and over and over again. But because of meiosis, every generation, natural selection has a whole different pool to sort through and pick out the fittest. And that's how evolution occurs. And we'll talk a lot more about natural selection and evolution later. And when we do, I'll refer back to, remember meiosis. Meiosis is one of the main sources of genetic variation. And the other one, just by comparison for right now, the other source is mutation. We talked about mutation in a previous lecture. So someone asked if we could go through the meiosis slides, so I'll do that. And so the first set of cell divisions is called meiosis 1. Meiosis 1 because it's the first of two divisions. At the top there, it says diploid. It starts with a diploid number of chromosomes, and at the end of the first cell division, it's haploid. So in interphase, the DNA has replicated. And you remember the 6 steps, the 6 enzymes that are involved in DNA replication. So those enzymes have done their jobs and they've replicated all the DNA and all the chromosomes during interphase. So then the cell starts to divide and it enters prophase. And prophase is one of the most critical points in cell division because that's where genetic variation is introduced. And it always is. In every meiotic division, you get genetic variation here due to crossing over. The amoeba sister told you what crossing over is. Or parts of chromosomes, homologous chromosomes. And homologous means one from one parent and the corresponding chromosome from the other parent. So they're not identical, they're homologous. And where they touch, they can exchange pieces. So in this case, we have blue and red. Blue can exchange with red with blue. In metaphase, so you go from prophase there, where crossing over can occur, to metaphase, where the chromosomes have to go to one side of the cell or the other. The important point about metaphase 1 is that the choice is completely random. So which side of this, the dotted line is called a metaphase plate. And any pair of homologous chromosomes can go on one side of that plate or the other. If they go on above the plate, they'll go to the top of the cell. If they go below the plate, they go below the cell. And that's entirely random. So it's called independent assortment. So remember those two terms. The first is recombination. That's the result of crossing over. And the other is independent assortment. And both of those are sources of genetic variation.
Can you write those down on the discussion, please?
On what?
On the discussion, on the chat or whatever.
In the chat, hang on just a minute. See if I can see chat. I'm going to have to get out of slideshow to see chat. Okay, 2 terms. One is crossing over. That equals recombination. And the other is independent assortment. So independent assortment happens in metaphase. And crossing over combination happens in prophase. Okay, can everybody see what I wrote in chat? And again, if you have any questions to address to Scott, please do that as well. Okay, share the screen again. And we're back to meiosis 1, the first division. So what we end up with to the right side of this diagram is two cells. Each is haploid, and that each has half the number of chromosomes as started with on the left side. But remember, each chromosome is actually two chromatids. And so the next cell division, meiosis 2, the point there is to separate those two chromatids so that they become full-fledged chromosomes. So genetic variation occurs in prophase 1 and metaphase 1. And now, are there any questions about this before I go to this? This shows you a close-up of how recombination occurs. You have sister chromatids, two red ones and two blue ones. They come into very close contact where they're touching each other, and that's called a tetrad, and that's where they exchange parts. So one of the red chromosome picked up a blue tip on one end and a blue tip on the other end, which leaves the blue chromosome with a red tip on one end and a red tip on the other end, and the other homologue continues to be blue. It has not undergone recombination. So this is how recombination occurs, and it is it's one of the sources of genetic variation and random assortment in the next step, where these chromosomes have to decide which side of the cell to go to. That also, because it's entirely random, it's a source of variation. So here's meiosis 2. Go back, these two cells here on the right, we go forward to the next step. There they are again, now on the left, and they are haploid, and they're going to continue to be haploid throughout meiosis 2. So we picked up these two cells in telophase 1, and now we go to prophase 2. And metaphase 2, where again, the chromosomes line up at the metaphase plate, and again, they can randomly assort. You can see the two possibilities here. And then in anaphase, they go to the poles, and then cytokinesis, well, I should say that after metaphase, the centromere is split, so that now the two chromatids of each chromosome become independent chromosomes on their own. So now these two chromatids go one direction and the other direction, and you end up with these 4 cells, which are haploid. They have one copy of each of the chromosomes, either red or blue or red blue or blue red. And you notice that they're all different from each other, genetically different from each other, and each one is genetically different from the parent. So this is a major source of variation. Every meiotic division, every time there's a fertilization, I should say, anytime that the testes and ovaries divide, have cell division, they produce a new crop. Each cell division produces 4 cells that are all different from the progenitor cell. Now, I sent you some worksheets, and I think that if we go over some of the, so the worksheets were all questions or exercises for you. So let me get out of this and call up a worksheet, and let's go through some of the questions. Hopefully, you've had a chance to try to answer the questions already. And is the print there big enough for you to see? Can you read the worksheet? Mitosis Meiosis worksheet?
Yes.
Okay. It's not showing up as a slideshow option, so we're going to have to go with what we've got here. So I just posed a series of questions, the likes of which might be on a test, either the one that's coming up next time on Wednesday, or some other test down the road. But for example, what is the difference between mitosis and meiosis? Now that's a very vague, open question, and you can come up with quite a number of things, but it's something for you to think about. And we've already gone over this. In mitosis, you have 1 cell divides to yield 2 cells. In meiosis, 1 cell ends up with 4 cells. In the case of mitosis, there is no genetic variation. In the case of meiosis, there always is. The purpose of mitosis is to sustain growth, development, and repair, whereas the purpose of meiosis is to generate, to reconstruct the genome, generate variation. And this provides raw material for natural selection over the course of evolutionary time. So in a nutshell, those are the basic differences between mitosis and meiosis. And make sure you understand that. It's important that you know why we have these 2 different types of cell division. True in all eukaryotes, not true in prokaryotes. Prokaryotes don't do either mitosis or meiosis. Second question, what is a karyotype? We went over karyotypes before. It's a visual display of chromosomes. What good is a karyotype? Well, you saw what can happen if you just count chromosomes. You can see that there can be abnormal numbers of chromosomes. That can lead to a change in phenotype. Or you may see altered chromosome morphology, which might indicate a problem. What is well, a change in karyotype, a gain of a chromosome, is called trisomy in a diploid organism. And what's the consequence of trisomy? For most of them, of the 23 chromosomes, most of them result in death. So the fetus never develops. There is no birth because the fetus aborts prior to birth. And a couple of trisomies, in the case of a Down syndrome, a person can live a relatively normal life. And there are other trisomies involving the sex chromosomes, which essentially have no change at all in the adult. Okay, I asked the question, mitosis is essential for growth, development, and repair. What happens in mitosis that contributes to these processes? Well, one is that the DNA is faithfully replicated without any changes. Copying has to be accurate, and it's equally distributed to the daughter cells. The number of cells is doubled with each division, and the production of new cells provides opportunity for differential gene expression. So remember that every cell has exactly the same number of genes, exactly the same genes, but they're not all expressed at the same time. In most stages of development, only a few genes are being actively expressed. And so as you have more and more mitotic divisions, each one, high fidelity, always producing exactly the same, you have an opportunity for a cell to receive a signal and differentiate into a specialized cell, like skin or lungs or brains, all the different kinds of cells we have in our body. So that's what happens in meiosis and results in us being able to be multicellular organisms. Okay, the next question is a matching. In and around the interphase nucleus, several structures can be seen if you look in a light microscope. And I've listed some of these structures on the left. And I said, match them with their functions from the list on the right. And this is typical of a question you might see on the test. So, nucleolus. We talked a lot about the nucleolus in the previous lecture. Does anybody remember what the nucleolus does? It's indicated there. It's item C. It makes ribosomes. So remember that. We have nucleolus and we have nucleus. They're very different from each other. The nucleolus is a suborganelle within the nucleus, and it makes ribosomes. The ribosomes, as you remember, leave the nucleus, go to the cytoplasm, get involved with the endoplasmic reticulum, and make proteins. So the endoplasmic reticulum here is D. It's where mRNA, tRNA, and rRNA all get together, collaborate, and make proteins. What are ribosomes? Ribosomes assemble proteins. They're like little machines that assemble, stitch amino acids together into polypeptides and proteins. What is a nuclear pore? Well, it's just a hole in the nuclear membrane where ribosomes can exit and get into the endoplasmic reticulum, which is sitting right there just outside the nucleus. What is chromatin? Someone asked this last time. Chromatin is chromosomes but they don't, it doesn't look like chromosomes because the strands are long and relaxed. When you look at them in a microscope, they're indistinct. You can't really see the chromosomes. Everything just looks fuzzy. And then during cell division, they shorten so that you can actually see that they are chromosomes. What is cytoplasm? It's the interior of the cell. It's thin, it's watery, and it contains all the different organelles that exist in a cell to perform all of the cell's functions. And what is a nucleus? It's the organelle in the cell that contains the genetic material, contains the chromosomes. So remember these relationships. They will come up, maybe not exactly in this form, but you need to know what each of these terms means. Then I asked, list the sequence of phases that occur in mitosis. So I've listed them here, and we've already gone over them in the slides. Interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. And at each one of these, a distinct set of activities. So know what they are. You need to know what each of those 6 terms is and what happens in that phase. What is the function of the centromere? You remember the picture of the chromosome with the centromere in it? Centromeres are usually toward the center of the chromosome, and they divide the chromosome into 2 arms, a P arm and a Q arm. And it's the point where microtubules attach. And the microtubules, what either pull chromosomes away from each other, or if it's in meiosis, chromatids. So the centromeres have to split. Remember that, centromere has to split to allow the chromatids or chromosomes to get pulled apart. In anaphase, this separation always occurs in anaphase, whether it's meiosis or mitosis. How does cytokinesis, which is essential to actually make 2 cells at the end of a mitotic or meiotic division, how does that happen? In the case of plants, there's a cell plate. In animals, there's no cell plate because there's no cell wall. And instead, there's a contractile ring. Another point that I emphasized is haploidy in humans. And one point that's important to keep in mind is that the haploid phase in humans is not prominent. And it can be, in terms of time, it can be very short. So meiosis occurs, and you get these haploid cells. Meiosis occurs in either the sperm, in the testis to make sperm, or in the ovary to make eggs. And you end up with these haploid cells. And so what's the difference between males and females during their haploid phases? Well, one is obvious. Females make eggs, males make sperm. In both cases, they're called a gamete. Females are born with their gametes. They're immature at birth, but the cells are there. Whereas boys have no gametes when they're born, no sperm, not even premature sperm. Girls start shedding their mature eggs. They mature 1 egg at a time, starting at puberty, and one egg matures and is shed every menstrual cycle. Now boys don't start making sperm until puberty, but when they do, they make huge numbers of sperm, whereas the number of eggs that females have is quite limited, roughly 500 mature eggs in the course of a lifetime. So boys start making sperm at puberty, and they can continue to do so to one extent or another pretty much throughout their life. For many men, gamete production never stops. Whereas once a female has shed all her mature eggs, that's menopause, and the reproductive life phase ends. So keep these differences in mind. They are important for you to remember, and could ask it. I'm not sure yet about the test on Wednesday, but there will be tests after that as well. The next question is a fill in the blank. The result of a mitotic event is to double the number of cells which are identical to each other and identical to the progenitor. I keep saying that over and over and over again because you need to remember it. That's mitosis. Double the number of cells, they're identical. Meiosis is very different. Instead of one cell generating two, as in mitosis, one cell generates, fill in the blank, 4, of which how many are identical to their parent? None. Each is different. Now, what are the sources? Yeah?
In meiosis.
In meiosis, you end up with 4 cells. They're all different, and they're all haploid. Okay, here is another important point, the next one on the list. Environmental conditions on the Earth are continuously changing over time, especially over geologic time, long spans. Living organisms must adapt as the environment changes if they're going to survive. Now, to do this, the genetic makeup of every species must continuously change over periods of time. What are the sources of genetic variation that allow species to change for natural selection to operate on variation? Well, they're listed here. One is mutation. We talked about mutation extensively, all the different types of mutations that there are. And a mutation can lead to any number of different phenotypic changes. But the other 2 terms here come from meiosis, recombination and random assortment. These are the terms I wrote in the chat. Recombination is when homologous chromosomes exchange parts with each other. Random assortment is a source of variation because when chromosomes go to the center of the cell and have to decide whether they're going to one end of the cell or the other, they really don't make a decision. It's all random chance. They just happen to end up on one side of the so-called metaphase plate or the other side. So that random assortment is a major source of variation. Any question about that? Sources of genetic variation in essentially all eukaryotic species. All right, the next question is true or false. Crossing over is physical contact between chromosomes of opposite parents and the exchange of chromosome parts. It's true. And finally, in meiotic metaphase, chromosomes line up the center of the cell. We've just been talking about this. And half of the chromosomes go to one side and half go to the other. What determines which side they go to? It's chance alone. It's not really determined at all. It's just chance. And that process, that chance determines where a chromosome goes, is called random assortment. Again, a term you need to remember: mutation, recombination, and random assortment. I sent you this worksheet along with several others, and this one I sent as an assignment. I sent 3 others in an announcement, and so then the purpose of these is to elaborate on the material in meiosis and mitosis, because there is a large amount of information. It's very important in the understanding of biology in general. And so that's why I'm spending 2 lecture periods on this topic alone, rather than going on to the next topic in this class period. So are there any questions or comments? Again, you have this worksheet. You can go back and study it in preparation for the test on Wednesday.
What was recombination again?
Recombination is when the chromosomes, and these are homologous chromosomes, exchange parts. And so in the illustration in the slides, you had a blue chromosome and a red chromosome, and where the blue and the red contacted each other, the red chromosome ended up with 2 blue tips, and the blue chromosome ended up with 2 red tips. Now, the exchange doesn't have to be at the ends of the chromosomes. It can be anywhere in the chromosome.
Professor, if I may ask a question?
Yeah.
So on the last answer, would you also accept independent assortment?
Yes.
Okay. Thank you, sir.
Yeah, that's a good point. Independent assortment is a term you will frequently see. So you should be aware of that and be able to equate random assortment with independent assortment. All right.
I had a question.
Yeah.
For the worksheet with the matching on the mitosis and meiosis, there's one of them, it says 90% total of the cell cycle time is spent in this phase. I was having a hard time finding that. What phase is that?
Okay, good question. I'm going to stop this and call up a different worksheet. Can you see this worksheet?
No.
All right, I have to do something different here. Now, can you see it?
Yes, sir, it's visible.
Mitosis, meiosis, cell division. You can't read it?
It is visible. Yes, it is.
Okay.
Yes, sir, we can see it.
In this worksheet, which is just a set of terms and their definitions, one of the terms is cell cycle. And so I put a diagram of the cell cycle here. And yes, you're right, you should study this and understand what it is. So most of the cell cycle does not involve mitosis or meiosis. The M, the little yellow segment of the cell cycle, is where cell division occurs, either meiosis or mitosis. But G1 stands for gap 1. And that's a period of time where the cell appears not to be doing anything, but it's actually checking itself for size, for its availability of nutrients, growth factors, whether it needs repair. It's considered a resting state. And then there's an S phase, that's synthesis. So that's when the 6 steps of DNA replication occur during the S phase. DNA is duplicated. Then there's another resting phase called G2 or gap 2, where the cells are checked again for size and make sure the DNA is replicated and replicated properly. And usually if mistakes are made in DNA replication, the cell aborts before it tries to divide. And then you have cell division as a small part of the cell cycle. Now, that point you made about G2 being 90% of the cell cycle, I'm not sure that's true. So don't hang your hat on that number. What you can say is true is that the cell cycle, excluding cell division, is 90% or more of the time. So a cell cycle for most body cells would be probably a number of days. Scott, you can correct me if I'm wrong about that. The synthesis.
It's highly dependent on what kind of cell.
Yeah, I'm sure.
For instance, they say the scent cells in the nose, I think, replenish every 120 days.
Okay. Okay. The DNA synthesis part is a matter of a couple hours for most cells. I'm sure that there's variation there as well. But DNA replication of all the DNA within the cell can happen in just a couple of hours. And then cell division, again, it occurs in a fairly short time. Hours, in the case of some cells, it can be minutes.
Would you accept the answer of interphase, as that typically includes G1S and G2 phase and G0?
Yes, that's a good point too.
So you could say interphase is 90% of the cell cycle and mitosis/meiosis is 10% of the cell cycle?
Yeah, roughly.
Roughly, of course. It's all approximations.
Yeah, yeah. The point is that proportionally, cell division is a very small part of the cell cycle. The rest of the time, the cell is just sitting there, checking on itself, or doubling its DNA. So the cell cycle is a 4-stage process. I'm reading off this sheet. In G1, the cells, excuse me, cell size increases. In the S phase, DNA is copied. In G2, the cell prepares to divide. And in M, you have mitosis or meiosis. Okay, we don't have a lot of time left. Again, this worksheet was sent to you in an announcement. So you can go over all these terms and look at their definitions. They're all given here. I want to stop sharing this and go to a different one.
Professor, I actually couldn't find that worksheet that we just went over right now.
It was not in an announcement?
No.
Okay, so
I
that's,
have 3 worksheets.
what are they?
Definitions of terms, circles overlapping, and the 10 matching.
Okay, so you're missing the definition of terms. I'll resend that in another announcement. Yeah, and that's important because it does have the diagram of the cell cycle in it. Okay, so I will do that. And you had the first one, was sort of a simulated quiz. You have that one. Did we just?
Professor, I'm sorry, I found it. I found it.
Oh, you did
It's
find
here.
it? Okay.
Yeah.
Then I can cross that off my list already.
Okay.
Great. Was there another question? All right, I wanted to go through this one with circles. I suspect you cannot see that at this point, but let me share the screen. Hope you can read that. The directions here place each word or phrase in the list on the left into either the circle for mitosis or meiosis, or if appropriate, in the intersection of, of the 2 circles. So each of these terms is either one or the other or both. So let's just start at the top with the time we have left and go down this list. Okay, 2 cells result. That would be mitosis. Next, 4 cells result. That would be, of course, meiosis. Increase in genetic variation of the cells. That would be only meiosis. Does not happen in mitosis. It's a 1 division process, mitosis, 2 division process, meiosis. DNA replication occurs in interphase. What would that be? Both. Remember, there's interphase for both of these processes. And that's where DNA replication occurs actually prior to interphase. But for purposes of cell division, we call it interphase. Produces gametes. That's easy. Only meiosis produces gametes. Produces somatic cells. Only mitosis produces. Somatic means body cells. Gametes means sex cells. Occurs in eukaryotic cells. Both. Mitosis and meiosis both occur in eukaryotic cells. Now, occurs in prokaryotic cells. I've highlighted that because it doesn't fit on this scheme at all. It's neither mitosis, meiosis, or both. In prokaryotes, it's not even called cell division, it's called binary fission. It occurs in prokaryotic cells. Can't have mitosis or meiosis because those are nuclear divisions, and prokaryotes don't have nuclei. So binary fission has neither mitosis nor meiosis, and it occurs in prokaryotes. It's an important point, and it's a distinction that you should have in your mind. Somatic cells, mitosis only. reproductive cells, meiosis only. Crossing over and recombination, that's genetic variation. And so where does that occur? Only in meiosis. Produces genetically identical cells only in mitosis. Sexual reproduction, you know that, it's in meiosis. Homologous chromosomes pair, meiosis only. Sister chromatids separate at anaphase in both. Asexual reproduction, mitosis. Chromatin condenses into visible structures. That occurs in both mitosis and meiosis. Haploid, meiosis only. Diploid is in mitosis, and we're talking here about the end result. The end result of mitosis is 2 diploid cells. In meiosis, it's 4 haploid cells. And then the last 3 terms, metaphase, telophase, and prophase, occur in both. So I thought this was a handy way of distinguishing between these processes and identifying what's common to both of them. And again, this should be in one of the announcements. And if you like, I can send you, in fact, I think I should, whether you like or not, send you the answers. I sent you sheets without answers so that you could look at them and try to answer them yourself. But now that we've gone over them, I'll send you the answer sheets in an announcement. So consider them study aids to prepare not only for the test on Wednesday, but any comprehensive test subsequent to that. Okay.
And you said the test is Wednesday, correct?
Yeah, Wednesday. So an important point. There will be no class period, no 12:30 class on Wednesday because the test could occupy as much as the full class period. I've designated 75 minutes for it, and it will be available all day on Wednesday, starting at midnight and ending at midnight. So you can take the test anytime during that interval that you like, but once you start it, you have 75 minutes to finish. And it is closed book, and it is monitored by Proctorio.
Thank you.
Uh-huh. And why don't you take this one down? And ask if we're out of time. So hopefully everyone has remembered to sign the chat for attendance. And Scott's workshop starts at 2. If you have questions you'd like to ask him or things that you'd like to just discuss from a different perspective, please attend his workshop. And yeah?
How many questions will be on the test?
Not quite sure because I've started it. It's not complete yet, but roughly 20, at least 20, maybe a little more than 20. And it'll be the same kinds of questions. Because I put such an emphasis on mitosis and meiosis, there'll probably be a preponderance of questions, but not entirely for sure. And So the material to be covered starts all the way back with transcription and translation, protein synthesis. So that's the starting point, and then everything that we've covered since then, which includes, don't forget, cystic fibrosis and pancreatic functions. There'll be something on that. There will be questions about mutation. And coronavirus. Don't forget about coronavirus. A couple of questions, I think, on membranes and transport. And as I said, several questions on mitosis and meiosis. The same kinds of questions that you're accustomed to. So some true and false, a couple of essays, but the essays are very short. Multiple choice, some fill in the blanks. Matching. Matching is a good way to ask you to make associations.
Sorry, I missed early. Did you say there would be anything about diffusion and osmosis or?
Yes, yes.
Okay.
I think I have already made up a question about diffusion. There might be something about tonicities. Remember, hypertonic, hypertonic, isotonic.
And what category would that be under?
Membranes and transport. Okay. If there's nothing else, you can always send me an email or attend Scott's workshop or write something on Scott's discussion board to get any answers that you need prior to Wednesday, prior to the test. And because the test is available all day, you can even continue to ask us questions during the day on Wednesday, as long as you haven't taken the test already.
So, Professor, I would just ask if you could hang out a second, and I could talk to you once everybody's left.
Yeah.
Before you shut down the Zoom. Thank you.
Okay. All right, everybody else, scram. Class is over.
Very amusing.
Yeah, that was a joke. I didn't really mean that I don't like you, because I do.
I just wanted to make sure that you didn't just end the meeting out. I wanted to give you a quick chat.
Yeah, okay.
So first thing, apologize that I was a little bit late there. Dr. Alberti had some questions for me after her class.
Mm-hmm.
And then I don't know what happened to my internet connection. It dropped out for about 10 minutes, but it seems to be working fine afterwards.
Yeah, I can hear you fine.
Okay, good.
Amir and Yosef, could you log out, please?
Or if they've stepped away, you might just have to disconnect them.
Let's see.
All right, sir. We free to go?
Yeah, please do.
All right, cool. Thank you.
Uh-huh. Yosef, let's see.
Is that on the three little dots that are on the?
Yeah, I just
upper
clicked.
right?
Yeah, I'm leaving. I'm leaving.
Okay.
Thank you so much.
Okay.
I hope you didn't feel like kicked out, but
No, just you and me now.
yeah. So I've been looking over the test, and I don't have a whole lot of feedback for you on it, but I will continue to look it over. I just wanted to let you know I got it this morning, and I've been reading through it during the lectures that I've been sitting at, and I was I had a question on it, and I was going to ask you real quick, but hopefully they got something out of those videos. You know, I know that's something different for you, and a little bit of a stretch to kind of probably feel like you're dumbing it down like that, but hopefully that was helpful to them.
I hope so. They're minimally responsive, and so it's just really hard for me to see what they're picking up. Now you see more of that in your workshop. You can tell from the questions that they ask whether they're way off base or close.
Right. And sometimes it's both ends of that spectrum. Oh golly,